Welcome to all 31 of you students in the audience today. I'm Rebecca Carter. I am the program manager of the STEM Lyceums After School Club, and we are with the Center for Excellence in Education. You're here joining us for our session, Opportunities in Research. Woo! Something you all want to get into in the STEM background. So welcome. It's great to see you. I hope you stick with me for the rest of the fall and spring season this year. And together we can learn more about STEM career pathways and college opportunities. Cool beans. All right, so before we jump into our program, we're going to go over some of the things you could know about Center for Excellence in Education. So first, CEE was founded in 1983 by Joanne DeGennaro and the late Admiral H.G. Rickover, who is the father of the nuclear navy and civilian uses of nuclear power. If you were with us last week, we did a session hosted by Sharon Smoot, and we talked about pathways in nuclear engineering. So shout out to that. So, our mission here at CEE is to nurture high school and university scholars to careers of excellence and leadership in science, technology, engineering, and mathematics, or STEM, and to encourage international collaboration between and among leaders in the global community. Here at CEE, we have four programs, the first of which is the Research Science Institute, or RSI, which has just wrapped in the last couple weeks. RSI is a highly competitive 6-week summer science and engineering program open to rising high school seniors. Juniors are eligible to apply. RSI is the first cost-free summer science and engineering program that combines on-campus coursework in scientific theory with off-campus work in science and technology research. RSI is paired with another flagship program, the U.S. Biolympiad, or USABO. USABO is the premier biology education and testing program for high school students. It provides the motivation, curricular resources, and skills training to take them beyond their classroom experience to the level of international competitiveness. Since CEE began administering the USABO in 2003, every member of the USA Biolympiad team has medaled at the IBO, which has just closed in Quezon City, Philippines. Shout out again, Blue Bullets, Ateneo. Our last two programs we have pair well together, the Teacher Enrichment Program, or TEP. TEP provides opportunities for middle and high school teachers to connect with leading experts from industry and academia, explore cutting-edge research and development, and make meaningful professional links with direct benefits for teachers and their students. Finally, you are here with me today, STEM Lyceums, a virtual club for high school students to meet once a month to learn about STEM career pathways from leading field experts. Industry professionals share cutting-edge research and offer unique learning experiences like scholarships, internship opportunities, and other opportunities for high achievers. We also offer other sub-programs such as the STEM Lyceums Mentorship Program and other programming towards college readiness, such as resume building and recommendation resourcing.
We will just start off with some pretty quick introductions from both me and Adrian. My name is Zain Zaidi. I went to high school in New Jersey, in the USA, because we have some international participants. West Windsor, Plainsboro High School South. I graduated last year. I just completed my first year of undergrad, and I'm going into my second year now. Um, I I go to Rutgers University. I'm a part of the Honors College, and so Rutgers University is in New Brunswick, New Jersey, in like central Jersey. My major is molecular biology and biochemistry. I just declared my major this past May, so I'm really happy about that. My minor isn't listed, but it's in Islamic studies, Islamic and Middle Eastern studies, sorry. And I'm on the pre-med track. I want to hopefully become a doctor one day. So my employment and internships, which I will go much more into depth with. I'm a research assistant at the Rutgers University Molecular Biology and Biochemistry Department. That's my major. I'm a clinical research assistant in two of our Rutgers University Med School's departments. There's one clinical research center and at the cardiology department. And then lastly, for further employment, I'm a patient care technician at St. Peter's University Hospital. So Robert Wood Johnson University medical school is Rutgers Medical School. Um, and so, yeah, the PCRC stands for Pediatric Clinical Research Center. and SPUH stands for St. Peter's University Hospital. These are all in New Brunswick, New Jersey.
Hi guys, I'm Adrian Perono. I also went to high school in New Jersey, but I went to high school at the Marine Academy of Technology and Environmental Science. I graduated in 2024, and that high school was a tech school that had a big focus on research. So I was really exposed to research like pretty early on in high school, like you guys, which is really nice. I'm also currently in my undergrad at Rutgers in the Honors College too. I have the same major as Zen, and I'm also on the pre-med track. For my employment and internships, I'm just recently finishing up my internship at Hackensack Meridian Health Center for Discovery and Innovation, which I'll go more into depth later. And I'm also a patient observer at Hackensack Meridian Health's Southern Ocean Medical Center.
Okay, great. So that was just a little bit about us. Now I'm gonna go into a little bit, or not a little bit, a good amount of depth into my research experience coming out of my first year of undergrad, and then I'll give you some takeaways. What did I learn from my first year of trying to get into research, being in research, um, and yeah, and then Adrian's gonna do the same, and then we're gonna wrap it all together with our most important takeaways from everything. We're gonna throw a good amount at you today. But you are welcome to ask questions in the chat. I will keep the chat open so I see them. Yeah, I'm happy to interrupt myself so I can answer any and all questions. All right, so let's jump into it. So my first research experience at Rutgers started with the Aresti Research Center. Aresti is, well, it's called the Aresti Research Center for undergraduates. It's the main channel by where Rutgers undergraduates can be involved in research. And so this isn't just biological research. This is robotics research. This is engineering research, computer science. There's all kinds of things that people are involved in. And so I'm gonna be honest about all my experiences. I'm gonna tell you everything beyond what the website will. So if you look up RSD, you'll see a good number of things, but I'm gonna talk to you about my experience. So there are two main programs for undergraduates as a part of RSD. There's a research assistant program and a summer science program. The program that I completed this summer, just 3 weeks ago, about is the Aresti Summer Science Program. It's about 30 PIs, which are principal investigators. These are people with PhDs who are experts in their field and they do research. And it's a program that matches up undergraduate students who are interested in research with these PhDs. And so I took interest in it just because honestly, it's just a popular program at my school. And yeah, there's it has a great history. It's been going on for 20 or so years. But again, I told you I'm going to be honest about my experience. Um, this summer, I really wanted to try to break into some more clinical experience. I told you guys I was pre-med, so I wanted to break into clinical experience, more delve into what it means to be a pre-med. And I did want to do more things than just my lab research this summer, though I'm really passionate about it. I wanted to split my time a little more evenly. So these summer programs, they're offered at many, many schools across the country. And if you are interested in doing other things than just research in the summer, this isn't really something I would recommend because I was trying to delve into my career as a pre-med, but this particular program, it was not every single day and you couldn't really take time off. Um, but yeah, that being said, I'm not like hating on this experience. I found it very valuable. I got paid, which is something that a lot of undergrads try to strive for, and it makes financial sense to get paid for the summer instead of doing nothing. I did research that I was actually genuinely passionate about. I enjoyed the people I worked with in my lab. I really appreciated my PI who helped me through everything. And I learned a whole lot. It's 10 weeks of full-time research working under a PI. Um, and yeah, it was great. And it culminated in the Aresti Summer Science poster session. So this poster session was for everybody who was in this program. I'd say it was about 40 kids, and then other programs also had their final poster presentation in the same room. So it was a bunch of people standing in front of their posters, telling whoever was interested what they did this summer. Um, yeah, and so the main thing I got out of this program were connections. Rutgers is a really, really big school, and it's easy to kind of once you meet somebody, it's easy to, never see them again. So I made valuable connections. I met a really great friend that I know I'm going to be friends with for a really long time. And yeah, some great personal and professional connections I made from this program. So this is my lab PI. I do lab research and I'm involved with clinical research. But because of the audience breakdown, I think we're going to be most interested in hearing about our lab research experience, which is what Adrian and I were kind of thinking in terms of organizing this. So anyway, this is my lab PI. The Rutgers University Honors College has this program called the Peer Mentor. No, sorry, Faculty Mentor Program, and it does what it sounds like. It connects undergrads, brand new freshman undergrads, to faculty members at Rutgers University based on common interests and stuff like that. Excuse me. And so he studies this stuff. It's a lot of words, but it's gene regulatory mechanisms. It just decides what genes are getting turned on and what genes are getting turned off in a cell. The specific cell he studies are neurons, and the specific neurons he studies are olfactory neurons. So these are the cells that are involved in smelling. He works with mice, and as you can see from this rather gory picture, um, I also worked with mice this summer. I did a lot of dissecting, um, yeah, and stuff like that. If you are looking to become a private investigator in your future, so I know a lot of people who get PhDs. Well, I don't know them personally, but I know a common route for people who get PhDs is either work in an industry or they decide to open up their own lab. And so this is a great route. I mean, these people are so knowledgeable and passionate about the research, and it's really commendable what they do. Um, However, from what I've seen, it looks to me like you're running a business. You're writing grants all the time. You're searching for money all the time, especially in our current, in the United States' current academic environment. Getting funding is not so easy. So it does look a lot like running a business than just doing the science that you love. It is a lot of that too, but you will have to diversify the time that you have if you do end up going that route. So now I'm going to talk about what I do, what science I do actually. Um, so to get into Kevin Monahan's lab, we matched up through the faculty mentor program. And so the first time we met, and it was kind of required because of the program, it was required that we had a meeting. So we had a meeting, we got some coffee together, and he told me he actually has no space for undergraduates in his lab. He told me that straight up because I think he sensed that that I wanted to get in. But he told me that. And his lab is pretty full. He runs a lab of about 10 people. So he told me straight up, I have no space for undergraduates in my lab. From the first meeting, however, I took interest in the science that he does. We discussed it at length for about like an hour and a half to 2 hours. And I just really liked it. The way he was able to explain his research to me was in a way that I could understand it, but also in a way that opened up so many questions for me in my mind. And I think that's one of the most important parts about asking questions. It's you have to understand something to a degree to which you can start to ask questions about it. So after our first meeting, I asked him even not to get into his lab in the future, just to meet again, just to talk, um, just to learn more about what his lab did, and just learn more about biochemistry in general, I guess. Before joining this lab, my original major was not going to be molecular biology and biochemistry, but after joining his lab, after learning so much from Dr. Monahan, I switched my major to MBB. So this is his lab background. I'm going to take you through what his lab does all the way through what I specifically did in the summer. So so he studies mice. So just keep that in mind. This is the context in mice. So we're looking at olfactory neurons, and olfactory neurons are the things that help us smell. So when a chemical from like a flower or a skunk travels all the way to a mouse's nose, it enters the nose and it hits an olfactory neuron. A neuron is a type of brain cell, and an olfactory neuron is the specific type of brain cell that helps you smell. Excuse me. So, what's interesting about these olfactory neurons, however, is that per cell, only one olfactory receptor is expressed. An olfactory receptor is a protein on your cell that picks up that chemical from the flower or skunk. So these receptors are proteins, and proteins are expressed by genes. Those are DNA. So DNA turns into proteins, and the specific thing about olfactory receptors is that only one active olfactory receptor protein is chosen for expression in an olfactory neuron per cell. So we have roughly 500 olfactory receptor genes in our genome. But per olfactory neuron, only one of them are picked for expression. So, as you can see here, every other olfactory receptor gene that isn't being expressed is clumped up together and doesn't get expressed at all. But the one olfactory receptor that is expressed is in this kind of hub. So let me get more into that. Um, a lot of this stuff is actually taken straight from my poster presentation that I did at the end of the summer. Um, and so this is what I'm presenting to you all right now is kind of a snippet um, of what my poster session looked like. So a common way that people present their research is not just presenting it in a written research journal, it's by doing poster presentations, sort of like this one. So in a mature olfactory sensory neuron, all that inactive OR genes are clumped up together. I showed this to you all on the last slide. And the one olfactory receptor OR that is chosen for expression is in this kind of hub. And this hub is formed by these two proteins, EBF and LHX2. The binding of EBF and LHX2 to an enhancer region is just a region of DNA that enhances, as the name suggests, the expression of another region of DNA. So when these two proteins bind to an enhancer, they recruit or bring along other enhancer regions to form this kind of hub. So as you can see here, this circle is a hub formed by the binding of these two proteins to enhancer regions, and then those enhancer regions, um, multiple of those enhancer regions, sorry, are recruited to form this hub. So this one active OR can be expressed out of a repertoire of hundreds. So these proteins, they bind specifically to a composite motif. A composite motif is a recurring sequence pattern that's present in DNA. This particular composite motif is the binding sequence where proteins, LHX2 and EBF, bind to the DNA. And it's called the motif. This motif is roughly 20 base pairs long, and about 81% of the enhancers that have to do with olfactory receptors contain this composite motif. So, oh, I'm sorry. So similar to mice, human olfactory neurons also express one OR per cell and form enhancer hubs like this one right here. However, many human OR genes are now non-functional due to mutations. By examining the conservation of Greek island enhancers and their composite motifs between mice and humans, we can determine if this OR choice mechanism is also conserved. I won't get into what a Greek island is, but it's basically just a name for the enhancers that enhance the expression of olfactory receptor genes. So, and this is also pretty complex molecular biology. But I mean, I find it pretty interesting. I hope you all do too. If you have any questions again along the way, don't hesitate to let me know. So I discussed this composite motif. And so I discussed this composite motif with Dr. Monahan throughout the summer. He taught it to me the way I'm teaching it to you all. And that opened up questions for me. Um, how did this composite motif and how did this mechanism evolve? So he was telling me this is the way it works in mice. I had a question, the question that I came up with that is going to drive my research personally for the next 2, 3 years is how did this research mechanism evolve? Now, a lot of undergrads, they don't come up with their own personal research idea. That's not the common route that people take. But I just had this question. Dr. Monahan said that actually hasn't been studied yet. Let's study it. And so we did. We started in the summer. This is a plot that I created in the summer. It looks pretty simple, but it took me like a month to just make this plot. I did a lot more bioinformatics work than I previously thought I'd be doing. But it turned out to be a great learning experience and I gained a lot from it. So this particular plot is a comparison of the presence score of the composite motif between mouse and human for each Greek island enhancer. So Greek island enhancers are the enhancer regions that enhance the expression of olfactory receptors. Excuse me, they exist in both mice and humans. And for each of these enhancer regions in both mouse and human, we wanted to see if the composite motif was present in these enhancer regions. So we saw that 12 out of the 63 enhancers, these OR enhancers that have so far been found, um, 12 of them did not have the composite motif. And so what that means is that there are these enhancers, and they just do not have this composite motif. They don't have this pattern. And so for those 12 that showed to not have that specific pattern, we took an idea from another paper. So a lot of what research is taking ideas from other papers and using them, using their specific protocols to see if they can be applied to other research to help us out. So we did that. Another paper discussed that regulatory regions, one type of regulatory region is an enhancer region. Um, they studied enhancer regions and they saw that they're evolutionarily conserved between two different species if anchor genes are also conserved. So what does that mean? What does conservation mean? Conservation means that if there's a set of genes that's present in mice, and it's also present in the same pattern or organization in another species, you can say that's conserved across evolutionary time. So mice and humans do share a common ancestor way, way millions of years back, but somehow through the process of evolution and evolutionary conservation, these genes in the same pattern remained. So that's evolutionary conservation. Anchor genes, however, are genes that surround our genes of interest. So this is the Rhodes enhancer region. This is one of the enhancer regions. There are 63. And out of those 63, 12 did not have the composite motif. So for those 12, we performed this kind of analysis. One of those 12 was called Rhodes. It's the Greek island Rhodes. And it's flanked by these two random proteins. Oh, somebody raised their hand. Um, these anchor genes are NDUFA10 and COPS9. They mean nothing to me. They mean nothing to my research. But what is cool is that these two genes appeared next to each other in the mouse genome, and they also appeared right next to each other in the human genome. Between these two genes in the mouse genome, there was a cluster of olfactory receptor genes. These are genes that code for those olfactory receptor proteins. And the same thing was present in humans. There was a cluster of olfactory receptor genes. So we thought that was pretty interesting. This is pretty much proof that evolutionary conservation does exist between the mouse genome and the human genome, specifically in terms of olfaction and olfactory receptor genes. One of the, I mean, so there were 12 out of 63 enhancer regions that did not have that did not find a match. They did not find a sequence match. Five of those 12 did turn out to have a match using this kind of analysis, this anchor gene analysis. But for those last 7 out of 12, we saw that there was just no match. There was nothing there. There's nothing that indicates, basically, from our preliminary search for Lyros and Mykonos, other enhancer regions, um, that there's a match between human and mouse. As you can see from this track here, it's kind of hard to see, but this region, this is an enhancer region found in mouse. And if you look at human, it's blank. That means that region is just completely lost. And there's no evolutionary conservation for that region between mouse and human. So this was just the beginning of our research project. This is just what we did in the summer. And our future research that is likely going to take until I graduate. I am a rising sophomore, by the way. It's going to take another 2, 3 more years to determine why aren't these regions conserved? What happened between the mouse and human lineage that made these two genes lose their enhancers? Did humans, and then we started to ask questions. So you can kind of see at the beginning, I asked a question. We did some experiments. These are bioinformatic experiments, not necessarily lab experiments, but biology nonetheless. We did some experiments and they left us with some more questions that we need to ask and answer. And we're starting to develop some hypotheses about these questions. So, why were these regions completely lost? Some of the hypotheses we want to test in the future are maybe humans developed something completely different that's unique to them. I mean, humans and mice do have a common ancestor, but they are still pretty far apart on the evolutionary tree. Did they develop something that's unique to the human genome or the human lineage? And there's another possibility is just that humans don't need to smell as much as mice do. Humans have trichromatic vision and mice have di or bichromatic vision, which basically means we can see a lot better. So mice rely a lot more on their smell, whereas humans can rely a lot more on their vision. And so that might just be a reason why 7 out of the 63 of these enhancer regions were completely lost. Um, I actually just explained this stuff to you, but most Greek islands, these enhancers are sequence conserved. It just suggests that the binding of those two proteins that I showed to you in the beginning is a conserved feature of these enhancer regions in humans. But several of these Greek islands have lost the motif. And for those that have lost the motif, that's what we want to continue doing research in. So I know that was some pretty heavy-hitting molecular biology. Uh, but to wrap up my lab research, our future direction for this is to study species between mice and human. So if mice are here on the evolutionary tree and humans are here, there's a whole bunch of species in between, like tree shrews and great apes and stuff like that, that we want to study their genomes that have common ancestors between mouse and humans. We also want to collaborate with other researchers at Rutgers to start doing some actual procedures. And having established this approach, can we determine how far back in evolutionary time can these enhancers go? So this it might even take us to amphibian olfaction, like tadpoles and stuff like that. How do they smell, and how do their 3D genomics organize themselves in a way that only one olfactory receptor is expressed out of a repertoire of hundreds. So that was just my lab research, and it's definitely not necessary for you guys to know what molecular biology I just explained to you, but this is the important stuff. This is what I would want to know if I was in high school. So that was my lab research. I do clinical research as well. In my freshman year, I have been doing, well, I did clinical research in one department at Robert Wood Johnson, and now I shifted to another department at Robert Wood Johnson. So the one that I joined first was the Pediatric Clinical Research Center, and they were doing a study on COVID-19 on a, actually a different disease called long COVID. It's basically prolonged COVID-19 symptoms after a person is originally infected, and we're studying how that disease impacts children. So that's what I was studying at the Pediatric Clinical Research Center. I started in September 2024, and same at the Department of Cardiology in 2024. I did city trainings. City trainings are basically just they're regulatory things. These research institutions want to make sure you have the proper trainings done to make sure you're not gonna leak their data or do something terrible to their research institutions. And I got IRB approval in November for the Pediatric Clinical Research Center. IRB is the Institutional Review Board. That's just Rutgers allowing me, hey, you're cleared. You don't have any conflicts of interest to join this research. I used my city certifications from the Clinical Research Center to boost my resume to get into the Department of Cardiology. And so at this point in January 2025, I am a research assistant, technically clinical research assistant, in both of these departments. Um, from January to February in this CRC, I was doing patient visits, which was something I really valued as a pre-med student. I got to see my own patients. I got to do their blood pressures and temperatures, which I thought was really cool. I was having a lot of fun with that. But it was patient visits for like 2 months. At the same time, in the Department of Cardiology, however, um, I wasn't included in anything. Uh, so for clinical research, a PI is actually just a doctor, an MD. And so at the same time in January in the Department of Cardiology, I was doing nothing but having meetings with the head doctor of the research team. He invited me for meetings, uh, and then nothing would really come out of those, even after follow-up emails or nothing would really come out of it. So as you can see, later after around February 2025, I really wanted to get myself involved in a research study. I wanted to get myself involved in a paper, um, whatever. So I set up meetings with the director. I sent countless emails. I joined every possible team meeting, and that goes for both departments. By the time January 2025 came along, at the CRC, I had been folding boxes and constructing office chairs and office tables for a few months. Adrian's my friend, and he knows that I would come back from this CRC office just complaining about how I'm doing no science, I'm doing no research, I'm an office intern. However, by the same time, in the Department of Cardiology, I gained Epic access, which is electronic health records. This is how we can analyze patient information so we can use them for clinical research studies. Now, from July to August, I haven't gone to the Pediatric Clinical Research Center. I completely gave up. They, I mean, I didn't feel like I was being treated well after almost a year being a part of their office. I didn't feel respected. I didn't feel like my input was valued. And their culture of work was very much isolated. This summer, however, in the Department of Cardiology, I am involved in three different clinical research studies. One's related to atrial fibrillation in patients who take anticoagulants with a disease called cardiac amyloidosis. There's another study that is involved with the screening for cardiac amyloidosis in South Asians. And my last study is about stroke survivors and hearing their stories as a part of a clinical research study. All of this is to say that you can be a part of however many labs you want to be a part of. You can get yourself involved in clinical research you want to. But if their culture of work and their culture of collaboration is either toxic or non-existent, it's not worth your time. And it's something that you have to get yourself out of as soon as possible. I was involved at the CRC for about a year until I finally decided to cut my losses and just leave. I figured folding boxes wasn't going to do much for my resume. And no matter how your mentors encourage loyalty in a given office, you do have to end up cutting your losses because if you're staying loyal with somewhere for a year and they still have you folding boxes and it doesn't seem like you're making any intellectual or professional progress, I would say get out. However, in the Department of Cardiology, at first I felt like I wasn't making much progress. You know, I started and for months there was nothing, but sometimes you just have to be persistent. It's about balancing persistence versus cutting your losses. Um, so yeah. Lastly, to finish up, appreciate the pace of your environment, and understand the activity of your office despite funding losses. Funding losses are very prominent in research in the US. And never double book yourself. This is something I did in the Department of Cardiology. I had a meeting with a doctor who was very, very busy. I double booked myself. I wasn't able to attend the meeting, and he almost didn't want to work with me, but because of my persistence, he gave me a second chance. And my last thing that I'll discuss is the pop-in. Last week, actually, how I got involved in the screening in South Asian study was I was in the office. I was at the hospital one day just because, and I decided to pop into the doctor's office because I just wanted to see if he had a free second to talk to me. He did, and I got myself on another study. So yeah, I'm sorry. I know I went over time. Adrian, I will let you, excuse me, take over. No. Sorry, I have one more slide. Um, so cold emailing, I that's how I got into the Pediatric Clinical Research Center. But as it turned out, it wasn't that great of a position. It's valuable. It's the way most students say they got research, but it's not the only way. If you want something, don't be afraid to just ask. Do the pop in, do whatever you need to do. Just ask. Ask a question. People love to answer questions more than you'd think. Embrace unexpected tasks and challenges. I thought I'd be doing a lot less computer work and bioinformatics in my lab, but I ended up really liking it and learning a lot. And ask for clarification and repetition. Sometimes PIs can ramble on and on about molecular biology or what have you for 20 minutes before they ask you if you have a question. Don't be afraid to interrupt them, stop them, and ask for clarification. They will always be receptive to it. Um, for any and all questions that come to mind. Okay. Adrian, I know I'm sorry I went over my time, but
No, you're good. I'm gonna share my screen now. Okay, so now I'm going to talk about my research experience in my undergrad. So, like I mentioned before, I am finishing up my internship at the Center for Discovery and Innovation, and this is Hackensack Meridian Health's medical research institution. Right now, they have 32 principal investigators, and their mission is to develop and rapidly translate innovations in biomedical sciences to better prevent and treat life-threatening diseases. Now, when I talked to the CSO of Center for Discovery and Innovation, his name is David Perlin. He told me that when he was recruiting PIs, his main goal was to make sure that the PIs were contributing to research where it would have a real patient impact. So the four main areas of focus for CDI are infectious diseases, cancer, immunology, and neurocognitive and behavioral health. So I joined CDI through the Summer Scholars Program. So in this program, students work under a PI and we just contribute to the lab. High school and college students above the age of 16 are eligible to apply. So if you are in the North Jersey area, I really do encourage you to apply because this is a really wonderful experience. This is a picture of me working in the lab, and then this is a picture of my lab group. So the PI I matched with, his name is Dr. Alvin Makahon-Moore. So he studies cellular and environmental factors that shape tumor evolution. And he also studies evolutionary dynamics by using experimental evolution of cancer. And I know that sounds kind of crazy, but I'll break that down in the later slides. So some background. The Makahon-Moore Lab uses evolution as a framework for investigating cancer because um, every phase of tumor evolution has evolutionary processes. So for example, in the tumor, there are a bunch of different selective pressures. So as a result, different cells with different mutations evolve. So this is kind of that idea of survival of the fittest, where cells with the most advantageous mutations will proliferate and outcompete other cells. But why does studying cancer evolution even matter? Why are we studying it? So the reason that cancer is so hard to treat is because cancer cells evolve and become resistant to treatment. And you can see that right here with this picture. So This was a patient with melanoma and subcutaneous tumors, which you can see in panel A. And he was given the vemurafenib treatment, and you can see that that worked in panel B. I mean, this picture was taken 15 weeks after the treatment, and you can see that those tumors are gone. But 23 weeks later, the tumors show up again, and that's because cells evolved and became resistant to that vemurafenib treatment. So this is just a schematic of the Makahon-Moore Lab workflow, but the overall goal of our lab is we study cancer evolution so we can target the evolutionary mechanisms and prevent this treatment resistance that I'm talking about. So now I'm going to be starting to talk about our current project. Um, Sorry, I don't know why it's not like working. Oops. Okay, yeah, sorry. Um, so the cell line that we're working with right now is called H716, and this is a human colorectal cancer cell line. So this cell line was isolated from the colon of a patient with colon cancer. So if you're wondering what a cell line is, it's just a population of cells derived from a single cell that's maintained for extended periods of time. So the cell line, the H716 cell line, was established in 1984, and labs have been using it ever since to study colorectal cancer. Now, the main star of our lab is this machine called a bioreactor. So the bioreactor is a machine used to grow cells in a large volume in a controlled environment. So if you look at the different parts, that kind of glass vessel with the yellow liquid in it, so that yellow liquid is actually media, which contains cells. So this vessel is where the cells are growing. And on top of that, attached to the vessel, there are probes and pumps. And the probes constantly measure temperature, pH, and dissolved oxygen, while the pumps add nutrients, acid, or base to make sure that these levels are where they need to be. And there's also a stirrer that keeps the cells evenly mixed. And we use it in our lab, we use the bioreactor in our lab to culture cells long term so that we can observe cancer evolution. So we're recreating cancer evolution so that we can observe it. So our current project focuses on how baseline and excess glucose conditions impact colorectal cancer cell evolution. So in our project, there are two groups. In the first group, there are three vessels contain cells cultured in baseline glucose media, and the other three vessels contain cells cultured in excess glucose media. So what I mean by baseline and excess is baseline glucose media contains just the level of glucose that cells need to survive and like not anymore. But excess glucose contains more glucose than the cells know what to do with. So all six of these vessels are grown in the bioreactor for 6 weeks. So we can observe evolution. And weekly, we collect samples and record data to monitor the changes that occur over time. So now I'm going to get into what kind of data we collect and the tools that we use to collect that data. So the first thing is the CDEX bioanalyzer. So this is a tool that lets us figure out the concentration of the metabolites in the media. So using this machine, we can put in a media sample and we can learn the concentration of glucose, glutamine, ammonia, or lactate. Another thing we do every single week is slides. So we spin down a sample of cells onto a slide so we can observe the cellular morphology and how that changes week to week. Um, Another machine that we have in our lab that's really interesting is the Guava cell analyzer. So with this machine, you basically just put in a sample of cells and it can tell you a bunch of different information like cell count, proliferation, cell death, mitopotential, DNA damage, and oxidative stress. Another thing that we do in the lab is cell pellets. So we make cell pellets for each of the vessels and then we send off those pellets to get DNA sequencing and RNA extraction. A big part of this whole puzzle of cancer evolution is studying clonal dynamics. A clone is a group of genetically identical cells that have descended from a single cancer cell. So you can see kind of in this picture here. Clones, so clones share the same mutations. And clonal dynamics are how different populations of clones evolve and interact with each other inside a tumor. Now, how do we identify these clones? We identify these clones using the genetic barcoding technology, the clone tracer system. So in the system, a DNA barcode is a short, unique piece of DNA, and scientists insert a different DNA barcode into each cell. So cells pass this barcode when they replicate, so they'll pass it down to their daughter cells. So all the cells are barcoded, and all the cells with the same mutations have the same barcodes. And we use this technology to determine which clones are dominant and when. So my undergraduate takeaways. My first one is to reach out to labs early. We're going to go more into depth about this later, but definitely reach out to labs sooner than later. But at the same time, don't pick the first thing that you get offered. Make sure you join a lab that you're actually interested in, so you can really contribute meaningful work. Professors like to recruit freshmen because they can get more work done than a senior. Also, my second undergraduate takeaway is that a good mentor is really helpful. They can help you figure out what you want to do with your career. In my experience in my program, we met a bunch of different PIs throughout CDI, and some of these were translational research physicians. Translational research physicians are just physicians who also have a hand in the lab. So they take what they learn from the lab to improve their patient care. Um, and I talked with some of them because I thought this was something that was really interesting, and they were all really willing to help me on my journey. So most people are willing to help, you just have to ask. My third undergraduate takeaway is that there's really something for everyone. I know that me and Zen stayed in the realm of biological research, but there's literally a lab for anything you can think of. I have a friend who's in a political science lab. Also, look for something you're really interested and passionate about so it won't feel like a task. And if you're really passionate about it, you can do meaningful work. My fourth undergraduate takeaway, and this is the really important one that, like, if there's a takeaway that you're going to take away, it should be this one. It's to always actively pursue opportunities. So in your undergrad, opportunities don't just fall into your lap. You really have to advocate for yourself. So whether that be a cold email or whatever, you really have to be that person to make that first step. Um, for me, and this goes with the always be curious part, the reason I got to do this program is because, like I mentioned before, I'm a patient observer at one of the other Hackensack hospitals. And in our orientation, they mentioned CDI. And I thought, wow, that's really interesting. I wonder if there's a way I could get in on that. And I just went on their website and there was a application for the summer program. So if there's something that you see that you think is interesting, just try to get in on it because the worst thing that they could say is no. And if they say yes, it really opens up a world of opportunities. So now I'm going to talk about the benefits of doing research in your undergrad. So the first one is mentorship.
Adrian, I'm sorry. I'm sorry. Could I just interject?
No, you're good.
Um, there were questions that were coming in
Oh,
um
sorry.
throughout that I did not notice. So this is kind of like the second part of our presentation, right?
Mhm.
Okay. So let's answer these questions and then we'll finish the second part.
Okay.
Okay, cool. Um the and we can go like back and forth on these. Do you have those open on your computer?
Yes.
Okay. So are these groups exclusively for undergraduate students? So Adrian and I are in different programs. My particular program was meant for undergraduate students. I know that there are similar programs at a whole bunch of other universities that are just for undergraduate students. Um, but Adrian's program wasn't.
Yeah, if you're 16, you can apply to this program. It's in North Jersey, like I said before. So if you live somewhere close, make sure you look into that because it's a really great experience.
Yeah.
Another question that we got is how is lab research different from clinical research? And I think Zen can answer this question.
Sure. So lab research and clinical research, they're different mostly in the ways that you collect data. Lab research, you collect data through like actual experiments. You do a bunch of micropipetting, you knock out some genes in some cells, or you kill some cells, or you do some things to some things, and then you get your data from doing that. You can't do that so much in humans. You can't just knock out a gene in a human. There's a lot more ethical things that you have to go through for clinical research. Um, but similarly, what I've learned is that between clinical and lab research, a lot of it can be produced from already published sets of data. So the data that I was using this summer, I didn't do any actual like lab experiments to do my summer research. The same way that I'm not doing anything new, like a clinical drug trial, for my clinical research. We're using previously obtained and previously published data, and reformatting it and looking at it in a different way, so that it shows something new, and something that people don't already know. But there's also translational research that Adrian was talking about, that kind of blends the two. Next question is, what extracurricular activities were you involved in and how did they impact your high school experience? Okay, high school extracurricular activities. So, honestly, my high school extracurriculars were not really related to research. I had one lab research experience that was sponsored by my high school. Um, and yeah, but I did a lot of theater stuff and all that. But, yeah, in the other ones, they look be getting a little more specific. So at the end of the Q&A, we'll circle back to the.
But before we do that, I'm gonna answer that question too. I was actually involved in research in high school, and research in high school is a little different than research in college, just because research in high school, you are really it's independent research. You know, you have your own project. You're kind of like the PI there, and the pathway is kind of just going to the different science fairs in your area. But in your undergrad, you're working under a PI. So you're working under projects that they're interested in, under like their topics. Yeah. For my other like activities in high school, I did like a bunch of things. I played viola, I ran track. So yeah. But yeah, we're gonna get back into the benefits of undergraduate research. So the first one is mentorship, and I mentioned this before, but mentors are really helpful in guiding your career pathway, and the PIs that you're working under are really good mentors. Um, Another benefit to undergrad research is exploring your interests. So research really opens your eyes to different careers and topics that you might be interested in. Like before, I had no idea what a translational research position was, but it's like all I can think about now. And if you think about it, undergrad is the perfect time to get involved in research because undergrad is really that time that you should be using to explore your interests. Another benefit to undergrad research is boosting your resume. So, in a lot of applications for grad school and medical school, they're looking for your story and also a genuine passion. So, research can be a really important part of that story. Research experience also helps you stand out in general. Things like this, they love to see research because it shows critical thinking, problem solving, and like teamwork. And it also shows that you have an area of expertise that you really know about. And even if you don't go into science, employers like to see that you've handled complex projects because there's a lot of soft skills that you gain in research that can be applied to like anything. Something else is networking. So PIs can give you really strong recommendation letters. Like you can get really close to your PI so they'll know you really well. And they can also tell you about hidden opportunities like positions or programs that you wouldn't know about otherwise. And the last thing is making a real impact. And it's kind of easy to lose sight of this, but at the end of the day, you're contributing to a field of study that you're passionate about. And that in itself is really wonderful.
Yeah, if you could just go back so I can touch on this a little bit. Um, Boosting your resume is about including your previous experiences that would look specifically appealing to PIs. So resumes are used for applications and stuff like that, yes, but for future lab experience and growth and to that end, I recommend adding a specific section of your resume underneath each task that says techniques. I have it in bold and it says techniques and then exactly what I did. So I'm working in a super specific subset of molecular biology, but that's not what I'm going to write on my resume. I'm going to write that I'm working with, uh, I have experience working with mice, the types of experiments that I'm running because they're in common throughout a lot of things. Um, and yeah, making a real impact is that should also be going on your resume. Say I improved X, Y, and Z in my lab. And numbers are always great to have in a resume. Yeah. So unsure where to get started in undergraduate research. It's stressful. Most undergrads, at least at Rutgers, they might not even get into research until junior or senior year. But it is actually not that hard. You just have to stay curious about it, and stay trying, stay with it, because you will eventually get it. And there's a lot of places to look. You just have to keep looking at all the different places that are available to you or nearby, um, and just really try to reach out. So, university labs. These are like where I am and where Adrian is right now. These are just with PIs. They are sponsored by a university. His is sponsored by Hackensack Medical School, which is technically a university. Rutgers University, and a whole bunch of other universities, colleges. Um, Rutgers is also like big state schools like this are not bad places to attend for undergraduate because um, they do tend to have pretty big research funding and research institutions. But if again, research is something that you really want to pursue, try to tailor your college search to those kinds of schools. Clinical research that usually happens at medical schools, schools of medicine, um, or there are some private offices that might do it, but those are usually harder to find, and you'll need some more personal connections for that. And then summer programs. There are a bunch of different summer programs. Honestly, a great resource for me has been ChatGPT or different AIs, because they help me find different centers for research that I wouldn't have previously known about, instead of just having to Google it. Because Googling it usually gives me stuff that is either next to impossible to get into, or the application deadline is closed, or something like that. So use your resources as much as possible. Okay. Um, Adrian, it might just be me, but you might be super zoomed into the Google Slides. Um, okay, there, that's better. Um, so yeah, cold emailing is probably the most popular way that oh, it's super zoomed in again. Okay, that's good. That's better. Yes. Um, is probably the most popular way that people get into research. If I remember me in at the beginning of freshman year asking seniors and juniors how they got into their labs. How did you get into your lab? How did you get into your lab? 90% of them would say, I cold emailed. And cold emailing is just emailing a PI or a doctor out of the blue saying, this is my expertise, this is my experience, this is what I know. You'll attach your resume or something, and you say, can I be involved in this lab? Um, so yeah, cold emailing. Do you want to go over the 101, Adrian? Like how to do it? You're muted.
So when you're cold emailing, definitely include your resume. And this is probably one of the more important things. Mention specific topics in their research that interest you because like PIs love to hear when you're interested in something that they're interested in. Another thing is just keep it concise. These PIs are really, really busy and they don't have a lot of time. So if you're writing them like paragraphs on paragraphs, they're not going to read it. Like they don't have time. Um, but the main thing is to always follow up. Following up really increases your chances of getting into a lab because, like I said before, these PIs are really busy. So when you first email them, they might just not have the time. Like they might just be really busy at that time. But if you follow up and give them a reminder, that'll first of all show that you're actually interested in their research. And it's just another reminder for them to get back to you. So, yeah, if there's one thing that you should take away, it's definitely always follow up when you reach out to PIs. So, what is appealing to PIs? So, something that I mentioned earlier is that PIs like to recruit freshmen over seniors just because you can do way more in the lab as a freshman than you can do a senior. And you can see this in this graph. Like you can just get a lot more work done. And this is actually something that me and Zen's professor told us. Because when he was giving us advice into how to get into a lab, so this is something that's really important. And like I mentioned before, a genuine interest in their research. So how can you show that you're actually interested in their research? So when you're looking for different labs to join, all of these research facilities, they should have a directory of the different labs that they have. And then you can go into like click a specific one and then figure out like a summary of what they do and then figure out more in depth the science and what's actually going on in the lab. And when you're reaching out to these PIs, definitely mention these details that you're interested in because it makes you stand out. Yeah. Another thing is that PIs are not expecting you to have research experience when you apply for a lab. So if you're applying as a freshman, they kind of know you probably didn't do research in high school. So don't be afraid to reach out. Like these positions, these undergrad research positions are meant for you. You are an undergrad, you are qualified. So just reach out and see what happens.
Okay, so I think the last thing I'll talk about is just keeping track. Okay. Okay, there we go. So yeah, so if you're still undecided about what you want to pursue in your career, and you're in between multiple different ideas, maybe you want to go into engineering, maybe you want to go into medicine, it's always good to have different resumes that are tailored to those kinds of professors, PIs, or job recruiters, or whatever. It's a good idea to have one resume that's maybe more suited towards engineering, that is more computational, math-based, and you include more focus on research or experience through that. Or if there's something medicine-based, you include some more focus about your clinical experience or your scientific research. And so I just kind of like to keep track of all my old resumes. This is just a screenshot, but I have my old resumes going back to like 2022. Um, it's always good to just keep track of how the way you changed, the way you grew. How did you change as a person? How did you change as somebody who can market themselves? And how did you just grow professionally? Like, see what kind of stuff you were involved in back then and what kind of stuff you're involved in back now, not back now, right now. So how did your goals change and how did you grow and progress? Understanding that and understanding how that works makes for a great narrative in future applications. So something super important in med school applications is having a narrative, having some signs of growth, changing in thinking and learning. That's super important to have in med school applications, and keeping track of your past achievements um, is just great for you to see for yourself, but also something great for another institution to see. You can stay consistently aware of your achievements and what kind of things you are involved in. So if somebody you're networking with asks you what are the type of things you're involved in, you can answer them up front, right there and then. Um, yeah, so in your resume, include specific things. Numbers are always great. Obviously not too many unnecessary numbers in a resume, but include the number of hours, your specific publications if you have them, new projects, or the progress you're making. And yeah, cool.
So now I think we're gonna go into
Uh,
the Q&A portion. So if you have any other questions, you can just add them to the chat, but we're gonna answer the ones that people have already asked. So are these group no, we already did that one. Um,
Little to no research, but I'm interested in doing research in high school. Did we answer that one? I think we.
where is the best place to start? I think we kind of answered it, but yeah, just definitely look at the different like labs that are around you. You know, if you're in high school, they might not be receptive to really joining the lab, but I know at least at CDI, they're really welcoming to high school students volunteering in the lab, just executing different lab techniques. So just send out some cold emails, I guess.
And don't Yeah, and don't hesitate to reach out to a university near you. I know at Rutgers there are some labs that have high schoolers working there. It won't be for money and it won't be for credit, but it will be research. Illustrate what type of graph you used or data science methods you used to depict your results or conclusions. So I use this thing called RStudio. It works with R code. It's a type of code. And RStudio is something that I'm still trying to understand by myself, but some names of the programming softwares we use are called Tidyverse and R. R is just a type of code. I know Python works on RStudio, and yeah, Adrian.
Yeah, I mean, I didn't have any graphs on mine, so
Okay.
Another question is if you could go back and give advice to your younger self during the time in ninth and 10th grade as a high schooler, what would it be? I would definitely tell myself to take more challenging classes. I know AP Bio really helped me understand the lab background, but when I was first getting into the lab, because when you first join a lab, they'll have you read through a bunch of papers, and since they're like really, it's really academic writing, it can be hard to understand. So just challenging yourself early on will set you for success. So do you have
Yeah,
anything?
I think Adrian, you said it perfectly. The next question is, you mentioned that nerve cells only express one OR. Uh, one OR gene. Yeah, so one OR gene per cell. Why not express more? What happens if you genetically modify a nerve cell to express more OR genes? So my PI did actually do that so he discovered these OR enhancers. So he knocked out this entire mechanism that chooses one OR per cell. And what he saw was that the sense of smell kind of just is gone from these mice that he knocked it out from. The sense of smell is pretty much gone. The olfactory neurons, they don't mature past a certain point. Um, and yeah, I'm not really sure why. My PI is not really sure why, but that's the way it works. And if it works any other way, then smell doesn't really work the same way. Um, it might honestly just be because of sensory overload or like material overload. Producing proteins in a cell requires a great amount of material and genetic coding, so it might just be the overload from that. Okay, moving on. Could environmental exposure that humans are exposed to more could affect their olfactory genes or kill their cilia? So yeah, this is possible and it's a good idea, but our genetic coding for our olfactory gene mechanism is it's evolutionary and it's hereditary. So this was likely the same um, olfactory mechanism that was present in early humans, like Neanderthals and stuff like that, and they weren't exposed to the same environmental exposure we are exposed to now. They were likely exposed to the same things that mice are. So they're exposed to the same things, but they still have different mechanisms of smelling. So it's unlikely that it's environmental exposure. Okay. Adrian, do you want to talk about the data gathering process?
I think I kind of talked about the different tools that we use and the different data that we get, but we're in the process right now because we just finished up this project, so we don't really have any conclusions yet. But we're in the process of taking this data and analyzing it with stuff that you probably learned in your stats class, like ANOVAs and um, t-tests. So,
Yeah, pretty much same here. We use published data. There were papers about the stuff that we're interested in that had published data sets from bio stuff that they did in the lab. Um, so we're kind of just using that. I also use the UCSC genome browser. Take a look at that. It's actually really, really cool. We don't have time to go over that right now, but yeah. Could you please suggest better ways to get started with research? I'm very new and not sure how to get started. Um, if you actually want to email us, either one of us, we could help send you a few specific resources specific to your location and availabilities. Um, we went over how to get started with research, but if you want to get more specific, please feel free to reach out to us. Our emails are on the slide. Adrian, what conclusions did you draw from your cancer research?
yeah, I mean, we're still going through the data right now. I actually just like made a presentation, but I um yeah, I feel like this will take a really long time. So, but I'll just really summarize it really quickly. But we looked at the different how the colorectal cancer cells are processing glucose. And over time in the culture, you can see that the cells, when they have excess glucose, they're really diverting a lot of it to glycolysis or lactate production, which is interesting because that kind of process, it creates byproducts that the cells can use for proliferation. So, and with cancer, the big thing is the cells are proliferating really quickly. So that's kind of an interesting observation, I guess. Yeah.
Do you think it's worth it to go into research you need to pay for? Um, Honestly, generally no. Unless it's like, yeah, actually, generally no. Programs that you have to pay for don't really look good on applications or just look good for your resume. Getting into research that you need to pay for might honestly, I mean, it might be good preliminary experience that you should definitely look into. I look on Reddit for a lot of the programs that I am considering joining. So definitely, if for the specific program that you're looking into, definitely look on Reddit. Because that'll definitely say something, say if it's a scam or not, and stuff like that.
I mean, for me personally, I've honestly never heard of someone paying to do research. I feel like I've only heard of people like getting paid to do research. I know Zen had a stipend, but I mean, I was an unpaid intern, and a lot of PIs are really willing to just take on volunteers. So, yeah, just reach out to PIs, then try to do that for free.
Is the process different for international high schoolers? I guess it depends on if you're going to college in the United States. I had a friend who was international in high school, but he came to Rutgers University in the United States, and he found he was actually in my same research program. So it's not all too different. I know opportunities really started opening up for me once I had my college email, my zen.zaidi@rutgers.edu. That really helped professors. That helped me get exposure to professors' inboxes. Um, but yeah, otherwise, international high schoolers, while they're in high school, I'm not really sure.
Yeah.
Um, okay, meta-analysis on disease called Kawasaki disease, but some of the data sets are not publicly available. Do you recommend me? If you are trying to get public, like private data sets, um, it's possible to email the professors behind the studies, but unless you are working with the professor, unless you are backed by a university, it is unlikely that they were going to give you their data because, I mean, it's theirs, it's their intellectual property. It's unlikely that they're just going to give that away. Um, so yeah, for your study, I would recommend public data sets. Otherwise, you will need to reach out, backed by a professor or a university.
A problem that you might be running into is like sometimes you can gain access to these data sets with your like undergrad email. So yeah, maybe try doing that because I know if I tried with my personal email to get some articles, like it would not let me, but if you use your like undergrad email, it will. Um, Another question is if you try interning at a lab research center just to see how the process and work looks like, would that be possible, or is there another way to see how the research process looks like in person? I definitely recommend trying to intern at a lab, but if you kind of just want to get a sneak peek of what's going on, you can definitely meet with the PI and just try to arrange a meeting. I know Zen's done that like 1,000 times just to gain insight onto like prospective labs. But yeah.
Yeah, and maybe if you email saying I want to shadow you, see how you work, they might be receptive to that because a big reason that PIs aren't receptive to taking students on is because it takes a lot of time to train them and teach them about their concept. But if you say I just want to shadow you and learn from you while you work instead of like getting actually trained, they might be more receptive to that. But it's a matter of getting in their inbox and getting a response first. What classes did you take in college and high school that helped you with your research? Oh, Adrian, I know we have an answer for this one. So we took this class that has a big, long, scary name. It was specifically for students who had AP Biology credit. They took AP Biology in high school, and it was specifically also for freshmen. It was called Honors Introduction to Research in Molecular Biology and Biochemistry. It was honestly not. It was a good class. It t taught us a lot about molecular biology. It went on our resumes, and that was probably the reason both of us got our labs because of how much we learned from it. Um, so yeah, that was a really important class that we had to take that we took that I mean, we were really focused on getting our pre-med requisites done in the first few years of college, but this one class was kind of diverging from it because we thought it would be a good idea to get some research experiments if that's what we're going to have to do to get into med school. Um, so yeah, that was a good class. Some introduction to research in biology labs. If your school offers that, that would be good.
Yeah, I mean, that class really helped me understand kind of what was going on in my lab here. So the cancer lab is really molecular biology focused. So having that foundation really helped me and set me up for success. And it was also a really great experience because of the lab experience. So we got to do a bunch of different lab techniques in that class, and that really definitely helped us. Um, so then I think this question might be for you.
Wasn't the only way to find internship opportunities. What other ways can you reach out? So yeah, my lab. So honestly, it's about what Adrian said. Stay curious. I got my lab not by cold emailing. It was by joining this peer faculty mentorship program that matched students up with a member of faculty. Um, and yeah, that was actually how I met my PI, and we just started talking from there. Uh, just try to engage yourself in all of these, in as many different programs or mentorships that you can, even peer mentorship opportunities. I know STEM Lyceums has one for peer mentors. Um, so these programs are really good. They just are to help you get connections. And the last thing I'll say is stay curious. I know Adrian and I in our free time, while we were taking a break from studying, we'd just be scrolling on other people's LinkedIn's or other people's stuff just to see what are they up to and how can I make their opportunity my opportunity. It sounds a little selfish, but sometimes you just got to do what you got to do. Stay
Also
curious,
about
keep searching.
Yeah, sorry to cut you off, but it's just about getting inspiration, seeing kind of what opportunities are out there. Like that yeah, doing that really helped me figure out like what are the different things that I can do. Um, yeah.
I think
So
those are all the questions.
yeah.
Thank you for all those questions.
Yes, thank you everybody for all of those awesome questions. I know you probably have a lot more, but we want to make sure that we close out the session, that we give our thank yous and leave time for anybody that has to commute. So, on the screen right now, you can see Adrian and Zan's emails if you want to get in contact with them. I'll also share it when I send out the recording to you all. So before you all jump on out of here, please stick around for our last bit of closing because I'll tell you a little bit of our next session. Before we get there, thank you again to our fabulous speakers. It was awesome with great turnout. Thank you all students that joined us today. And again, thank you to our sponsors whose contributions let us have cost-free programming for you all. Especially thank you to the Claude Moore Charitable Foundation. Without our sponsors, we'd not be able to have all these opportunities for you all. And then moving on, please remember to fill out the post-webinar survey. This is what I use for attendance purposes. It also opens you up for opportunities like the mentorship program. And please register for our next session, STEM in the Environment. We're going to be talking about environmental sustainability, jobs in that space, and also the intersection between environmental sciences and the policy space. So if you're interested in that congressional opportunity, working on the Hill, and environmental sustainability, this is a really good opportunity for you. And I'll also email you the link when you get your recording. So again, thank you all for coming. Please fill out the survey and thank you to our speakers. Once again, it was awesome. You guys crushed it. It was great.
All right. Thank you, Rebecca. And thank you, everybody, for listening to our stuff.
Yeah, thank you so much for having us.